Hi, I'm Dr. Dan Davidson, Product Development Manager at Ag Authority. It is a pleasure to be here today to briefly present the poster evaluating the effects of carbon-based fertilizer on soil microbiome using PLFA. The objective of this study was to evaluate the impact of bio-based fertilizers on the soil microbiome using the phospholipid fatty acid test. This test measures and identifies the PLFAs found in a soil sample, offering a quantitative indication of microbial activity as well as the balance of different microbial groups in the soil. A corn trial was conducted at 4 locations in 2019 using conventional and bio-based fertilizers. Soil samples were poured weekly during the first 6 weeks, and PLFA, active carbon and nitrate, were measured and yield was recorded. An unfertilized non-crop reference check was included. ANOVA Test is a bio-based fertilizer with consistent analysis. The biophase binds nutrients to create a slow-release scenario that improves efficiency and reduces environmental loss. The project generated a large data set with 124 variables. That was reduced to 93 variables by imputation of the data, then further reduced by principal component analysis, 38 variables, and then later reduced to 9 variables by cluster analysis and correlation. Then after normality tests, those variables were analyzed by Kruskal-Wallis or ANOVA. This scatter diagram represents the principal component scores across the 3 X, Y, and Z axes. The 4 colors represent the different locations. The scatter plot reduced the variables from 93 to 38. These 38 are the most important. These 3 scores accounted for 51% of the variation in the original data set. Cluster analysis took the 38 variables and reduced them to 6 clusters and 9 representative variables that were either analyzed by Kruskal-Wallis, represented in blue, or ANOVA, represented in red. Variables shown in black are highly correlated and analysis would be redundant. The variables within a cluster have similar characteristics. The correlation analysis helped place variables into 6 individual clusters and assisted in identifying the 9 variables to analyze further. The bluer the color, the more highly correlated and redundant a variable. A variable had to have a coefficient less than 0.7 to be analyzed for normality. Table 1 lists the 9 representative variables. The ANOVA and Kruskal-Wallis analysis were performed on the selected PLFA variables based on the Shapiro test of normal distribution. ANOVA was ran on normally distributed data, and Kruskal-Wallis was ran on non-normal data. Table 2 shows the Kruskal-Wallis analysis of 6 PLFA variables. The fertilizer regimes were significantly greater than the reference check. However, there was no significant difference between fertilizer regimes. Table 3 shows the ANOVA analysis of 3 PLFA variables. The gram-positive, gram-negative bacterial ratio, fertilizer treatments were significantly greater than the reference check. However, there was no significant difference between fertilizer regimes. The 19.3 eukaryote category and 17.1 actinobacteria category showed a definite fertilizer response, with Simtrex being greater than the commercial fertilizer and commercial fertilizer being greater than the reference check. Table 4 shows the ANOVA analysis of active carbon, nitrate, and yield. Active carbon and nitrate were significantly greater in the fertilizer treatments than the reference check, but there was no difference between fertilizer treatments. However, Simtrex 10S had greater nitrate levels than the other fertilizers. There was no significant difference in yield between the fertilizer treatments. This summarizes the results. The reference check was always less than fertilizer treatments. Sites did differ from each other, but fertilizer treatment responses were the same. While there were differences over time, different treatment differences also remained the same. Our statistical approach helped interpret the large data set that accompanied soil health testing and identified 9 representative variables we could evaluate initially for statistical differences. However, there are an additional 30 variables we can pull into biological interpretation later. As our understanding of soil health and its indicators evolve, it is important to identify and narrow down the key variables to assess soil health. All fertilizer treatments showed a significantly greater PLFA values than the reference check. Simtrex fertilizer did cause a shift in the bacterial population, which is a metric of soil health. Based on yield, Simtrex bio-based fertilizer performed equally to commercial fertilizers, yet provides a more sustainable solution to growers. Thank you for your time today.